In this demo, we're going to talk about single-cell sequencing and how we can use EPI2ME to analyze Oxford Nanopore single-cell data. And I think it's been clear from the talks we've seen throughout the last few days of NCM that really EPI2ME workflows are ready to analyze your data so you can get faster biological insights. We heard from Megan Noonan yesterday about how she used workflow single-cell to analyze the kidney tubule data. It brought new biological insights. Because of our long reads, we can cover full transcripts. And I've got Brian with me from the applications team who's going to take you through a full demo of the EPI2ME single-cell analysis workflow. Before we do that, though, I'd like to introduce EPI2ME version 5.2. We launched Cloud yesterday. The team were really excited to get this feature out to you. So now we can do both local and we can do cloud analysis through our desktop application, EPI2ME. EPI2ME is available on macOS, on Windows, and on our devices and Ubuntu. Makes it really easy to execute bioinformatics analysis now in the cloud as well. So we redesigned the user interface. It's now much cleaner. And we've got resource management front and center. You can see your analyses, your system CPU, and system memory available. And now we have cloud, right? You can run it however you want. If you've got enough local compute, you can run the workflow locally. And it's the same set of settings to run the workflow in the cloud. It's just a simple click of the button to switch between the two. The analysis is exactly the same. We have the same workflows available local and cloud. It's the same settings. Really easy to execute your analysis in our cloud platform. You also see the progress like you do with local workflows. So we're really excited about EPI2ME. It's free for download now. You go to our website, labs.epitome.io, and download the new version. And that's all I've got to say about EPI2ME now. We're gonna head into our demo with Brian. So Brian Leyland is a commercial applications manager, and he's going to tell us about our single-cell workflow. Brian.
Awesome, thanks,
Thank you.
Matt. Okay, so we're going to talk about the workflow single cell for analyzing your 10x experiments with Oxford Nanopore sequencing. Before we dive into that, though, let's go over a little bit about the value of long read sequencing for single cell experiments. So if you do traditional short read with your single cell experiments, you're only getting one paired read in each transcript, maybe one exon or just the UTR even. Whereas with and that's missing, about 80% or more of most transcripts in the genome. With long read sequencing, you're getting end-to-end complete transcripts, and that allows you to extract a lot more rich information from your samples and your 10x data set. So that allows you to see new things that you couldn't see with short read, like look at alternative splicing, different isoform expression and different cell types or cell subtypes. You can even do downstream analyses to look at fusion gene identification. We heard a little bit of that from the plenary talk in the Q&A session just now. So here are some examples. Again, as Matt mentioned, we've seen a couple examples today already, but here are a couple more that I'll just highlight, showing how this isoform level data full transcripts can add to your experiments. In this case, they were looking at Arabidopsis. On the left, you can see how short read sequencing clustered the cells into this cluster 2 in orange here. But on the right, with long read full transcript information, you can differentiate sub-cell types within that cluster 2.1 and 2.2 as they labeled here. So you're getting finer grain, richer detail with these long read transcripts here. Here's another example. This is also from mouse brain. We've seen some other mouse data at this event so far today. On the left, you see isoform-specific clustering. And on the right, we see a spatial experiment. So there, we're looking at the SNAP25, 2 different isoforms in the midbrain of mouse. And you can see different expression of these individual isoforms with this long read data. Okay, so this is a little bit about why you would want to look at whole transcripts and isoform level data for your single cell experiments. How do you generate those data with Nanopore? So the way this works, you may have heard in the session earlier today with Rachel and Carly. I've given a brief review of that here on this slide. I'd encourage you to go look at that previous talk online if you haven't already. You can see that it starts with a 10x kit. So this is compatible with the 3', 5', spatial and multi-omics kits for the 3' expression part of multi-omics. And then the prep that you do is basically just adding on our adapters onto the ends of these molecules that you've already generated with the 10x kit, and then sequencing that typically on a Promethean flow cell. It allows you to get 100 million reads, even more than that in some cases, for a single experiment, which is plenty of reads to look at thousands of cells in a typical experiment. Okay, so now that we've reviewed why you would want to look at transcript level information and how you generate those data with nanopore sequencing, let's dive in a little bit to the workflow single cell pipeline. Again, this is one of our EPI2ME pipelines that you can access in the desktop app or via the command line, and this is what kind of an overview of what this pipeline does. So at its core, it takes your raw reads and does all the pre-processing steps and barcode and UMI identification and clustering that you need to do to generate a matrix. Same as you would generate from a short read experiment at the gene level, but additionally it also generates a matrix of expression counts at the transcript level. And this is an end-to-end pipeline. It's got a bunch of default parameters already set, so it's really easy to just download and get started with this pipeline and get all the way through these steps without having to deal with the barcode or UMI handling yourself. You can run this pipeline through the desktop application, which we're going to look at in just a second here. You can also run it via the command line. It's based on NF Core standards, so it's deployable to lots of different compute if you run it via the command line. And it's really the requirements for running either of these ways, via the GUI or the command line, are very minimal. So it's easy to get up and running with this pipeline and all of our other EPI2ME workflows in really just a few minutes. What are the key outputs of this pipeline? So it gives you really detailed QC information. It allows you to assess your experiment, look for any potential problems with your library prep that are leading to any ancillary products that may be unwanted. You get to see knee plots showing you the cell count from your experiment. And then in addition to that QC information, which we'll look at in more detail in a second, you also get all these raw files. So you get a tagged BAM that has all of the aligned reads and cell assigned reads, information about the barcode, UMI and barcode correction for any of those reads, and then these matrices. So again, on the left you get a cell level or a gene level matrix, and on the right you get a transcript level matrix. And the report also generates UMAP plots both at the gene and the transcript level. So you can get a quick look at your data and the clustering of your cells in the experiment right from the report as soon as you completed the pipeline. Okay, so with that, we're going to start a brief demo looking at how you would kick off one of these analyses in the EPI2ME desktop application, and then what the report would look like. So we're switching over here, and thank you, Matt, for helping with the demo. If you want to start a run, you would click here. You can see all the different EPI2ME workflows that are available to run either locally or in the cloud with those little icons. And for this, we'll scroll down to the single cell one. You select that there. It gives you a description of each workflow, which has some nice overview and detailed information if you're new to this type of analysis. And then you can launch the workflow from here. And you can either run it locally or in the cloud if you saw those two options there. Sorry, we'll go back a second. Yeah, right there. And so we'll just simulate kicking off a run for this demo. You'd select here. You're going to select input files that you need. The inputs for this workflow are your reads in either FASTQ or BAM format, and then a reference transcriptome. So you would select those files from your local machine at this kind of beginning of kicking off the workflow. And these would be on your local machine if you're going to run locally, and they would get uploaded as the first step if you're going to run in the cloud. Then there are a bunch of arguments that you can tweak for running the workflow. As with all the other EPI2ME workflows, there are sensible defaults selected for all of these, so you shouldn't need to choose too many different arguments here. But if you do need to, one thing that I really like about this desktop app is that it has detailed descriptions that you can expand, so you can learn more about all the arguments and options that you can change as you kick off a workflow, like workflow single cell. And then once you've selected all those optional arguments, you would click Run, and that would launch the workflow. This workflow will take, it can run as fast as like a few hours if you have it on a really fast local machine, or it could take, about 8 hours or maybe a little bit more if you have a lot of reads. So we won't actually run the workflow in this demo, but instead we'll open up a completed report so that you can see what the report looks like after the workflow has completed. So first you'll see if the workflow were running, you would see the progress, like Matt mentioned earlier. This one is complete, so all the steps are done. Very nice graphical display of the progress there, which you would also see if you were running locally in the raw Nextflow output. All the files would be there, and then you'd click on that tab to see the report. This pulls up a report. You can also get this report as an HTML file so that you can easily share with other collaborators or people in your lab. And the report at the top starts with some QC information. So you can see information about the read quality score, read length distributions. And then as you scroll down more, there's a table showing you the number of reads, and then number of reads and reads that have been mapped and assigned in the table below. And then these are some really key figures here. This shows you the number of reads that are retained at each step in the workflow. So the number of full-length reads, and then on down reads that get assigned to a gene, and then at the transcript level. And then the plot just below this is showing some of the potential artifacts, like TSO artifacts, things that are not full-length transcripts that have been identified, and if you use the defaults of the workflow, have been removed from the analysis. This is really useful because it can help you diagnose problems with your library prep, where your library prep might have gone wrong. And if this is a kind of a new plot to you, you can look, there's really detailed descriptions in these reports that explain what the plot is and how you might look at it and interpret it to understand what's going on with your library prep. So if we scroll down a little bit more, the final set of QC information, you see a knee plot showing you how many cells above threshold have been identified, and then information about UMI and gene saturation in the experiment, and sequencing saturation. So then down here we have UMAP plots. So these UMAPs are coming off of the matrices that are generated at the gene and transcript level. So gene on the left, transcript level on the right. I know the text is a little bit small for people in the room here. But these plots are also interactive. So you can zoom in. You can look, that's colored by average expression, normalized average expression per cell. So you can scroll through any and interact like that. You can also save these as images if you want to add them to a presentation or share them with a collaborator. And then this one, I believe, is a spatial report. Yeah, there we go. Nice little checker there. But yeah, you can save them as images to share. So you can see some of the spatial information. So if you've run the workflow with an argument that specifies genes of interest, this will show you expression, spatial expression of some of those genes of interest. There's a drop-down menu there, and you can select different plots of the different genes of interest. And this just gives you a really good way to get a quick look at your data right as it comes out of the pipeline without having to do any other manipulation or visualization. All right, so, and these are also interactive, of course. And thanks for your attention.
Thanks a lot, Brian. It was a great presentation showing the power of single-cell analysis in EPI2ME.